let us proceed to our learning activity number 10, which is entitled Hematoxylin and Eosin Staining Method. Hematoxylin and Eosin, or commonly known as H&E staining, is the most widely used staining technique in histopathology. As its name suggests, H&E stain makes use of a combination of two dyes, namely hematoxylin and eosin. This combination differentially stains various tissue elements and make them easy for observation. For our learning objectives, you are going to enumerate the reagents used in hematoxylin and eosin staining method, identify the function of each reagent used for the staining process, explain the theoretical principles of staining, and perform the hematoxylin and eosin staining method. The following are the needed materials for this activity. So you have slide-mounted tissue sections, staining jars and dishes, staining racks and trays, xylene, alcohol formol, 95% ethanol, hematoxylin, eosin Y, acid alcohol, ammonia water, dishwashing soap, cotton swab, and paper towels. Before we will proceed with our lecture proper, let us first define what is staining. According to the book, it is defined as the process of applying dyes on the section to see and study the architectural pattern of the tissue and the physical characteristics of the cell. Staining is possible because different tissues and cells display varying affinities for most dyes and stains used during the process, so that first, they become more visible. Second is that morphologic changes are more easily identified. And lastly, the presence or absence of disease process can be established. Now, I want you to remember this, everyone. Acidic components have an affinity to basic dyes, thus they are termed basophilic, and basic components have an affinity to acidic dyes, and they are termed as acidophilic. Now I'm gonna ask you a question: why do we need to stain? The most basic reason that cells are stained is to enhance visualization of the cell or certain cellular components under a microscope. The major importance of staining is to increase or improve visibility and also to study specific features such as the morphology. That includes the shape, the size, and identification, structural details, and as well as its composition. Let us proceed to the three major groups of staining: histological staining, histochemical staining or histochemistry, and lastly immunohistochemical staining. In histological staining, tissue directly interacts with the dye. It produces coloration of the active tissue component. It uses microanatomic, bacterial, and specific tissue stains. It establishes relationship of tissues and differentiation of nucleus and cytoplasm. In histochemical staining, chemical reactions of specific tissue substance, such as Perls-Crucian blue, which stains our hemoglobin, and periodic acid shape, which stains our carbohydrates. In enzyme histochemistry, it uses the reagent as a substrate and the color reaction as the end product. While in immunohistochemical staining, it utilizes immunologic and histochemical techniques, as well as polyclonal, monoclonal, fluorescent labeled or enzyme labeled reagents. Now let us proceed to the various methods of staining. So staining techniques might employ more than one of these techniques. Methods are not mutually exclusive, and the methods are named depending on the context. So you have direct staining, indirect staining, progressive staining, regressive staining, metachromatic staining, counter staining, metallic impregnation, and vital staining. So we will go over with each of these methods of staining one by one briefly. The first method of staining is direct staining. It is defined as the process of giving color to the sections by using aqueous or alcoholic dye solutions. In simple or direct staining, only one dye is used, which is washed away after 30 to 60 seconds prior to drying and examination. Methylene blue is a classic example of a simple stain. So this blue stain will color all the cells blue, making them stand out against the bright background of the light microscope. Shown in the picture is our methylene blue and staining our squamous cells of the buccal cavity. Next, indirect staining is the process whereby the action of the dye is intensified by adding another agent or a mordant which serves as a link or bridge between the tissue and the dye to make the staining reaction possible. By itself, the dye may stain only weakly, if at all. Let us define what is a mordant. A mordant serves as a bridge between the dye and the tissue. Shown here in the picture is a tissue mordant dye complex. So the mordant plays an important part of the stain. So no staining will occur if there is no mordant used. Potassium alum with hematoxylin or early hematoxylin can be used as a mordant, as well as iron in your Weiger's hematoxylin, grams iodine in gram staining, and heat in our Ziehl-Nielsen method or the physical mordant used in microbiology. Let us define an accentuator. Accentuator is not essential to the chemical union of the tissue and the dye, or meaning it does not participate. But the action of an accentuator is that it increases the staining power and the selectivity of the dye. So some examples of our accentuator are potassium hydroxide in the Loeffler's methylene blue and phenol in carbol-thionine and carbol-fuchsin. Next, we have metachromatic staining. So metachromatic staining is that it differentiates a particular substance by staining them with color that is different from the stain itself, as opposed to orthochromic stains. Shown in the picture is our toluidine blue showing mast cells. Toluidine blue is a basic thiazine metachromatic dye with high affinity for acidic tissue components, thereby staining tissues rich in DNA and RNA. The use of toluidine blue in tissue sections is done with the aim to highlight components such as mast cells, granules, mucins, and cartilage. So the color shift is always from a blue or violet dye to yellow or red stain, meaning that the color absorption shifts to shorter wavelengths, leaving only the longest wavelengths to be seen. This is believed to represent polymerization of the dye. The greater the degree of polymerization, the stronger is the metachromasia. Next, we have progressive staining. Progressive staining is the process whereby tissue elements are stained in a definite sequence and the staining solution is applied for specific periods of time or until the desired intensity of coloring of the different tissue elements is attained. So shown in the picture is our methylene blue and staining our squamous cells of the buccal cavity. Next, we have regressive staining. With this technique, the tissue is first overstained to obliterate the cellular details, and the excess stain is removed or decolorized from unwanted parts of the tissue until the desired intensity of color is obtained. Routine hematoxylin and eosin staining is the most common method utilized for microanatomical studies of tissues, using the regressive staining, which consists of overstaining the nuclei, followed by removal of superfluous and excessive color of the tissue constituent by acid differentiation. So you can use various reagents or chemicals in order to remove or decolorize the excess stain. So you can use water or alcohol, use of acids and oxidizing agents, acidic dye. You can use basic solution as a decolorizer, and then the basic dye, you can use acidic solution as the decolorizer. If both acidic dye and basic dye, you can use an alcohol, and the mordant also can act as a differentiating agent. Next is counter staining. It is the application of different color or stain to provide contrast and background. So shown on the picture, the smaller one is our methylene blue used in the Ziehl-Neelsen method. And then the bigger picture shows the renal cortex or the juxtaglomerular apparatus via the H&E stain. So the eosin serves as the counter stain. Next, we have metallic impregnation. The tissue elements demonstrated by colorless solutions of metallic salts. So shown on the picture are the neurofibrils and motor neurons in the gray matter of the anterior horn of the spinal cord. So the stain used is this silver impregnation or the Cahals method, and the commonly used stains are our gold chloride and the silver nitrate. Next is our vital staining. It is the selective staining of living cell constituents. We have two types of vital staining, the intravital and the supravital staining. In intravital staining, you must inject the dye into any part of the animal body. So it can be intravenous. The commonly used intravital staining are lithium, carmine, and India ink. Shown on the picture are the gravid proglottids of our Diphyllobothrium species. So please take note of the characteristic rosette-shaped uterus of the parasite, and it is stained with India ink. While on the other hand, supravital stain is a method of staining used in microscopy in order to examine living cells that have been removed from an organism. It differs from intravital staining, which is done by injecting or otherwise introducing the stain into the body. Shown in the picture are reticulocytes and our Heinz bodies stained with supravital stain using our new methylene blue stain. So this is a peripheral blood film under oil immersion objective. So the methylene blue dyes called nucleic acid stains or vital stains are used to differentiate and count these young red blood cells. Vital or supravital stains are dyes absorbed by the live cells. Then the young RBCs contain ribonucleic acid or RNA and are called reticulocytes when the RNA is visualized using our vital stains. Heinz bodies are inclusions within the red blood cells composed of denatured hemoglobin. So it is an indicative of oxidative damage to our RBCs. What is the staining method of choice for histopathology laboratory? Yes, you are correct. The answer would be Hematoxylin and Eosin or H&E staining. Hematoxylin and Eosin or H&E staining protocol is used frequently in histology to examine thin sections of tissue. Hematoxylin stains cell nuclei blue, while Eosin stains cytoplasm, connective tissue, and other extracellular substances pink or red. Eosin is strongly absorbed by the red blood cells, coloring them bright red. In a skillfully made H&E preparation, the red blood cells are almost orange, and collagen and cytoplasm, especially the muscle, acquire different shades of pink. When the staining is done by a machine, the subtle differences in eosinophilia are often lost. Hematoxylin stains the cell nucleus and other acidic structures such as RNA-rich portions of the cytoplasm and the matrix of our hyaline cartilage blue. In contrast, eosin stains the cytoplasm and collagen pink. Our H&E staining is first introduced by Boehmer and Fischer in the year 1865 and 1875 respectively. The principle behind H&E staining is the chemical attraction between the tissue and the dye. Hematoxylin, which is a basic dye, imparts blue to purple contrast on basophilic structures, primarily those containing nucleic acid moieties such as the chromatin, ribosomes, and cytoplasmic regions rich in RNA. An acidic eosin counterstains the basic elements such as RBCs, cytoplasm, muscle, and collagen in varying intensities of pink, orange, and red. Now let us proceed to our hematoxylin. It is extracted from the Mexican tree Hematoxylum campicianum. So hematoxylin extract is ripened to obtain hematin. Hematin is by far the most valuable staining reagent used by the cytologists due to its powerful nuclear and chromatin staining capacity and its striking polychrome properties which may be produced with proper differentiation. Corumbia opaca is also known as the desert bloodwood, is also a species of a tree that is endemic to northern Australia, which can also be used for H&E staining. Shown in the picture is our Hematoxylum campicianum tree. Hematoxylin can be ripened in two processes, the natural ripening and the artificial ripening. So for natural ripening, you can have an exposure to air and sunlight, but it is usually slow, for it will take you 3 to 4 months before the hematoxylin will be ripened into hematin. While in artificial ripening, it is accelerated by strong oxidizers such as hydrogen peroxide, mercuric oxide, potassium permanganate, sodium perborate, or sodium iodate, which converts your hematoxylin to hematin almost instantaneously by chemical oxidation. Let us discuss further about hematoxylin. Hematoxylin alone is not technically a dye and will not directly stain the tissues. It therefore needs to be used in combination with a mordant, in which we have defined earlier that a mordant is a compound that helps it link to the tissue. So mordants are substances that combine with the tissue and the staining solution, forming a bridge that allows staining reaction to take place. So you can use aluminum or alum salts, iron, chromium, and copper salts as our mordants. Hematoxylin in complex with aluminum salts is cationic and acts as a basic dye. It is positively charged and it can react with negatively charged or basophilic cell components such as nucleic acids in the nucleus. This stain blue as a result. Eosin is an anionic and acts as an acidic dye. It is negatively charged and can react with positively charged acidophilic components in the tissue, such as amino groups in proteins in the cytoplasm. These stain pink as a result. One of the mordants used to combine with our hematoxylin is our aluminum or alum salts. So it can produce a blue coloration to our tissues. examples of our aluminum or alum salts are the following. You can have Ehrlich aluminum or Ehrlich alum, Harris, Coles, and Mayers. So these aluminum or alum salts are examples of progressive or regressive staining methods. So the Ehrlich alum salts can be ripened by our sodium iodate. The Harris alum salts can be ripened by mercuric oxide. Coal, the oxidant is iodine, and the Mayers is iodine. You can also use iron or ferric salts, which can result into a blue-black coloration. Examples are the Weigerts or the Heidenhains, and it is using the regressive staining method only. So for Weigerts, the mordant is ferric ammonium chloride, and then for Heidenhains, the mordant is the ferric ammonium sulfate. You can also use 1% aqueous phosphotungstic acid, and usually the method employed is progressive staining. We have three types of our Eosin stain. So first is the Eosin Y, which is yellowish in color, and this is the most commonly used type of our Eosin. We also have Eosin B or erythrocin B, which is bluish in color, and it can cause a deeper red color of our tissues, and it is rarely used. And lastly is our ethyl S or the ethyl Eosin, which is rarely used also. So in the preparation of Eosin Y, you can add thymol crystals in order to prevent the formation of molds upon the preparation. This is just a review of our tissue processing steps. So first we have fixation, second is our dehydration. We also have clearing, paraffin infiltration, embedding, sectioning, and our staining. So the calcification also is an exception rather than a rule. Now let us proceed to the basic steps in staining. So we have the paraffinization, hydration, removal of mercury pigments, staining, dehydration, and clearing. Now we will go over with these steps one by one briefly. The paraffinization, it is the removal of wax, and it's done usually with xylene. It is essential to remove the wax completely, otherwise subsequent stages will not be possible. At least 2 to 3 changes in xylene are needed in order to ensure complete removal. So sections at this stage should appear clear and transparent, and the presence of any patches indicates the presence of wax in sections should be kept longer in the xylene. The next step is hydration. Xylene is not miscible with aqueous solutions that are used later in the staining process. Therefore, it is removed with graded alcohol from higher concentration to lower concentration. First, change is made to absolute alcohol or acetone, followed by 90%, 70% alcohol, and finally distilled water. Sections now should appear opaque. Presence of any clear areas are indicative of the presence of xylene. So in order to remove this, sections should be returned to absolute alcohol and rehydrated, or the so-called sections to water versus sections to alcohol. The next step is the removal of mercury pigments. So mercury chloride is the most common metallic fixative. Others are chromate and lead. Except for Heidenhain-Sousa, black precipitates of mercury are deposited in tissues because of those metallic fixative. So mercury chloride and other metallic fixatives should be removed first before staining. So in case of mercury containing fixatives such as zincer is used, mercury pigments are precipitated on these sections. It has to be removed before staining is done. Removal involves treatment with iodine solutions, which changes the mercury to an iodine compound. This in turn is converted to tetrathionate by thiosulfate, which is readily soluble in water. The slides are placed in running water to wash out all extraneous chemicals. The next step is staining. Various staining agents are applied during this hydrated stage. This involves exposing the tissues to hematoxylin, Eosin Y, and strong chemicals like acid alcohol and ammonia. Care must be observed during this stage to avoid detachment of tissues. Potassium alum will dissociate with an alkaline medium, forming insoluble aluminum hydroxide from soluble red to insoluble blue medium, thus resulting to bluing. 2 ml of strong ammonium hydroxide with 98 ml of tap water can be used, and then ammonia may be hard on a delicate tissue and it may loosen and cause sections to fall off the slides during staining. Next, we have dehydration and clearing. Dehydration is done using graded alcohols from 70% to absolute alcohol. Dehydrating alcohol can remove some of the stains, so time has to be suitably modified to minimize fading. Since alcohol is miscible in xylene, it is used for clearing the sections. Any sections from which water has not been completely removed would give a milky appearance after the first xylene. Such sections should be returned to absolute alcohol and the process is repeated. Mounting is done after second or third xylene. So there are 2 reasons for clearing. First is that most mounting media, especially resinous media, are immiscible in alcohol but miscible in xylene. The second one is to raise the refractive index of the prepared slide. Now let us proceed to the Hematoxylin and Eosin or H&E staining procedure. From the slide warmer, allow the slide to cool for at least 5 minutes. Then proceeding with staining, dip or immerse the slide in the following order of reagents. First is to clear the paraffin embedded sections in xylene for at least 2 changes and 5 minutes each interval. But according to the book, it can also be 2 to 3 minutes. Next is to immerse in the bath of absolute ethyl alcohol according to the book, but according to our lab manual, you can use alcohol formol for at least 10 dips. And then next is to transfer it to a bath of 95% ethyl alcohol with at least 3 changes, 10 dips each. Next step is to wash the slides in a soapy water and remove excess albumin or impurities using a cotton swab. Remember to not to touch the tissue sections, then rinse the slide well with a clean water for at least 1 minute and then drain the excess water on paper towels. Next is to stain the slide in a hematoxylin for 5 to 10 minutes and then drain the excess stain, then rinse with water. Next is to decolorize the sections in an acid alcohol. So you can take 2 quick dips only and then you must rinse the slides immediately in water. Next is to immerse the slides in ammonia water until tissue turns blue with an average of 5 minutes. or you can use 1% aqueous lithium carbonate in case ammonia water is not available, until the sections appear blue for about 30 seconds. Then after that, you must wash again in a running water for 5 minutes and then drain the excess water on paper towels. The next step is to stain the slide with Eosin Y for 1.5 to 3 minutes and then drain the excess stain, then rinse well with water. Then drain again the excess water on paper towels. And then after that, you must dip it in a 95% ethyl alcohol for at least 3 changes and 10 dips each change. And then lastly, you must dip the slides in saline, again 3 changes and then 10 dips each. And please don't forget to wipe the edges and the back of the slide to clean it, and please take extra care not to touch the sections. This is how the manual H&E staining setup looks like. After staining the tissue via H&E staining, you can now mount the stained tissue. And here's for our reference. So thank you so much everyone and have a good day.